Okay, great. Well, good afternoon, everyone. Welcome back to our COVID seminar series after a little break in February. Just a reminder, please turn off your video so that the quality of the talk will be optimal. Today, we're really excited to have Dr. Caius, Radu, and Tang Ting Wu, both faculty in molecular and medical pharmacology, to give us to tell us about their recent advances on mRNA vaccine research, which is obviously a very hot and exciting topic. Caius is a professor in molecular and medical pharmacology and surgery. He's also the vice chairman of the Department of Molecular Medical Pharmacology. He's also the co-director of Cancer Molecular Imaging, Nanotechnology, and Theranostic Research Program at the JCCC. He initially got his MD from University of Medicine, Craiova, Romania. Then he did his postdoc training at UT Southwestern and also at UCLA with our very own Dr. Owen Whitty. His group has been actively involved in advancing our understanding of biological processes at the interface between metabolic, signal transduction, and immune networks in cancer and other diseases, and to basically apply this knowledge towards development of new diagnostic and therapeutic approaches. And so obviously, I can imagine the COVID vaccine area is of great interest to his lab. After that, Dr. Ting-Ting Wu, she is an associate professor also in molecular and medical pharmacology. She got her BS degree from a National Taiwan University, followed by her PhD at University of Pennsylvania. She then did her postdoc training, again, in our very own Dr. Ren Sun's laboratory here at UCLA. And, and she's been, really excited about the idea of basically attenuating, the virus, right, to generate new vaccine candidates by removing the ability of the virus to counteract the type 1 interferon. And he has done collaborations in the past with Ren Sun's lab on using high throughput profiling methods to identify mutations that will make flu virus more sensitive type 1 interferons. And as a result of that, they found some really interesting and exciting vaccine candidate. And her research program is obviously very interested in developing vaccine strategies for herpes viruses, and also to understand the molecular mechanisms, how these viruses manipulate the host gene expression, and also developing virus-like particle vaccine platforms for these herpes viruses. So sounds like a really exciting duel. Please take it away, and thanks for coming today.
Thank you, Melody, and thanks everybody for joining. Today we will try to give you a brief overview of what we have done in the last, I would say, maybe 5 months in terms of mRNA vaccines. Just to now take a step back and then look at the timeline of vaccines. You can see on this slide that probably the first appearance of mRNA vaccines on the stage was in 2013, and those were preclinical studies. But they set up the stage for what most of you know, I think all of you know, has been an amazing development in 2020, which is essentially the development, the fastest probably development of vaccines ever, essentially starting from the design to initiation of clinical trials in less than 3 months. Now, this kind of gives maybe the impression that mRNA vaccines are something entirely new, but as I'm going to show you briefly, they have been in the making for decades. And in terms of names that you probably heard being associated with mRNA vaccines and maybe even candidates for a Nobel Prize in not distant future, I want to mention Drew Weissman and Katalin Kariko. Their work was actually instrumental in the development of current mRNA vaccines. And looking at one of their landmark papers, it was published in 2005 in Immunity. Here they introduced this kind of concept of modifying the mRNA vaccines so they can actually be used in vivo. Before this work, every attempt to actually, kind of inject at that time, of course, mice with mRNAs would lead into result in severe toxicity. So they had the insight of, well, kind of now at that time, the properties of mRNA in terms of activators of various pattern recognition receptors or toll-like receptors were becoming more well established. So they had the idea of modifying essentially the mRNA to attenuate their activation of pattern recognition receptors. And here are the modifications that they used. They took uridine and they converted it to 5-methoxyuridine, or we are going to hear much more about pseudouridine, but both are modifications that now render an mRNA unable to activate or significantly reduce the ability of mRNAs to activate two very important toll-like receptors that are toll-like receptors 7 and 8. And in terms of how does this look, this is taken from the paper. We are looking at activation of 3 different TLRs, TLR3, TLR7, and TLR8. All are RNA sensors, but of course TLR3 senses double-stranded RNA, whereas TLR7 and 8 sense single-stranded RNA, which will be, of course, like mRNA. If you look at unmodified mRNA, both TLR7 and TLR8 are strongly activated, but now if you modify uridine nucleotides, replace them with either with 5-methoxyuridine or with pseudouridine, now you see that activation of TLR7 and TLR8 is significantly abolished. So that opened the field for medical applications for mRNA vaccines. Now fast forwarding to 2020, you've seen maybe the cover page of Time magazine, and of course the key role played by other people, including Gugur Sahin, who is the founder and CEO of BioNTech. And of course these vaccines make a huge impact. Again, you have heard about this now is real world data from Israel showing that the BioNTech vaccine BioNTech Pfizer vaccine used in that country essentially abolishes the incidence of severe COVID-19 disease. However, kind of like there is, of course, still, there are still unanswered questions, and there is actually probably a lot of room for improvement. One of the areas that is of high interest to us relates, of course, to induction of T-cell mediated CD4 and especially CD8 T-cell responses by mRNA vaccines. So here we are looking at data with clinical data with the Mrna vaccine, and as you can see, there is some, of course, kind of some induction of CD4 T-cell immunity, but CD8 T-cell responses are actually not that impressive at all. And now the BioNTech vaccine works somewhat better, but in both cases, we think that there is significant room for improvement. And while this might not be really necessary in for the majority of people who are vaccinated with either the Mrna or the Pfizer BioNTech vaccine, there are situations in which the lack of insufficient CD8 T-cell mediated immunity can be a problem. And what we kind of think as a future direction where we can actually have, maybe there is room for improvement. For instance, in the ever-growing number of patients who have now been treated with CAR T-cell therapy with CD19, CD20, and as a consequence, they have severe B-cell aplasia, and they are incapable of mounting an antibody-mediated response. So in that case, you would like to have stronger CD8 T-cell responses, so to compensate for the lack of humoral immunity. That's basically what we are trying to do long term. So the objectives for the project, first, we wanted to help expand the mRNA vaccine program at UCLA. We strongly believe that that this is an area that will continue to grow, and UCLA hopefully will have a very strong presence. We wanted to achieve a better understanding of the adjuvant properties of mRNA vaccines. And of course, kind of like for those who know what my lab is working on, and as well as Ting-Ting's lab, it is not surprising that type 1 interferon will take the center stage there. And then maybe kind of now, as a consequence, we'll be able to learn more about how to tune the adjuvant properties of these mRNA vaccines in order to maximize CD8 T-cell mediated immunity against both viral and in the context of what we do in the lab, tumor antigens as well. So now what have we done so far? We started simply by learning how to design these mRNA vaccines, and we have 4 of these constructs. 3 of them have been generated already, and Ting-Ting will show you data for the first 2 constructs, that 1 and 2. The first being the RBD domain from SARS-CoV-2. Then we have the HA protein from the flu virus. A very important construct for us has been this firefly luciferase, which enable us to use bioluminescence imaging to monitor the pharmacokinetic properties of these mRNA vaccines. And there is another flu construct that is now in progress. So all of these constructs are modified in terms of their uridine residues have been replaced with methyl pseudouridine for constructs 1, 2, and 4, and with methoxyuridine for the 3rd construct, which is firefly luciferase. So now after we design them, the sequences are sent to a company in San Diego called TriLink. They clone them in their expression plasmid. They do the in vitro transcription. There are purification steps. Now here I want to note that constructs 1 and 2, the RBD and HA constructs, are not HPLC purified. From that point of view, they are identical to commercial mRNA in the Moderna and Pfizer BioNTech vaccines. And that is because at this point it's impractical to perform HPLC purification at a scale required to for these vaccines, and that's something that will be very important for the rest of the talk. The construct that it's on the bottom of this list here, the NP construct from the flu NP construct, that one we just order it, and that will be HPLC purified. Now, once we have these mRNA constructs, we have established an agreement with a company in Canada called Aquitas. You might have heard about it. They are world leaders in essentially packaging mRNA vaccines into their lipid nanoparticles. These lipid nanoparticles from Aquitas are actually used in the BioNTech Pfizer vaccine, and as well as in the CureVac vaccines. They are, I don't want to spend too much time, part of mRNA vaccine story. It's something that goes back 4 decades at least, and development of lipid nanoparticles has been probably the most challenging part, and continues to be one of the bottlenecks in producing more mRNA vaccines in a short amount of time. But now once we have the agreement with Aquitas, we can send them mRNAs, and then they package them and they send them back to UCLA, where we can store them at -80 degrees and in single-use aliquots. Now, I have to go now in a little bit more detail what happens, how an mRNA vaccine works. It looks like a complex scheme, but I'll try to guide you through it quickly. So, of course, we have a lipid nanoparticle in which a nucleotide modified mRNA vaccine is encapsulated. However, kind of like for all the current mRNA vaccines, they are not HPLC purified, and it's almost unavoidable that there will be double-stranded RNA byproducts of in vitro transcription that will also be included in the lipid nanoparticles. Now, once these are encounter antigen-presenting cells, especially immature dendritic cells, they will be internalized, and this occurs via macropinocytosis. they will end up in the endolysosomal compartment. There will be a change in the charge of the particle. They are ionizable particles. They become positively charged. They interact with the membrane of these vacuoles. They fuse with those membranes, and now they can discharge their content both in the endosome as well in the cytosol. In the upper part of the scheme, you see that once the mRNA reaches the cytosol, you'll have translation, and then you end up with the antigen that is encoded in that mRNA. However, there is a second process that happens in the same time, which takes place both in the endosome and in the cytosol, and there is strong activation of, depending on the amount of double-stranded RNA, of various pattern recognition receptors like TLR3 in the endosome, and RIGI and NBA5 in the cytosol. TLR7 and TLR8 will, in this case, not be activated because, as I mentioned before, these mRNA vaccines are nucleotide modified. As a consequence of activating these pattern recognition receptors, it's not only antigen that is being now expressed, but also a variety of cytokines, including type 1 interferon, which will be probably the most important of them in the context of this talk. Now, zooming out and looking at the subsequent steps, for simplicity, I broke them down. In the upper part of the slide, you look at the uptake of the LNP mRNA and its translation. In the bottom part, it's basically activation of pattern recognition receptors and interferon production. Now, a key role in this process is played by type 1 interferon, which is required to be generated in order to mature and induce maturation of dendritic cells and their migration to draining lymph nodes. And also, of course, interferon will help priming of antigen-specific T-cells. However, there are two other aspects that have to be taken into account. Of course, these mRNAs resemble viruses, and cells that take up the LNP mRNA particles will actually mount something that resembles an antiviral response, which again is mediated by interferon and leads to inhibition of mRNA translation. And that can be detrimental if the response is too strong, because then you don't have enough antigen being produced following the uptake of the mRNA. And the second component that it's less well understood, we think that activation of antigen-presenting cells also, and secondary to that of non-antigen specific T cells, can induce complex signaling, transcriptional and metabolic alterations, which can in the end modulate cellular immunity. So I'll tell you two stories about each of these two components, which kind of again illustrate the complex biology of interferon, the double-edged swords that the type 1 interferon, the roles that it plays. On one hand, priming immune responses, and on the other hand, being actually sometimes immunosuppressive. So the way that we are measuring the interferon production by mRNA vaccine, we simply use bone marrow-derived macrophages. We take them, generate them from mice, and once we have these macrophages, we can treat them with different mRNA lipid nanoparticles. And I'm going to show you data for the firefly luciferase construct and HA, the flu HA, and the SARS-CoV-2 RBD. As a positive control, we use a compound that activates another pattern recognition receptor called STING, which stands for stimulator of interferon genes. And we use a STING agonist to achieve that STING activation. And I'm going to come back and describe how STING functions and what happens when we use a STING agonist. After 6 hours, we can collect mRNA and then look for cytokine production. And that's what we see here. We see, in terms of interferon, a very robust induction of type 1 interferon by the STING agonist, so the positive control. The lipid nanoparticles themselves, without any mRNA, they are inert, so they don't have any adjuvant capacity. Our construct, mRNA construct for firefly luciferase, is also inert. However, the two constructs for HA and RBD, they induce very strong, they are very strong inducers of type 1 interferon, even stronger than the positive control. The same patterns can be seen for IL-6. So what we have learned from this very simple assay that the F-look firefly luciferase mRNA construct, which is the one modified with 5-methoxyuridine, it's immunologically silent in urine macrophages, and because it does not induce type 1 interferon or IL-6. On the other hand, the HA and RBD mRNAs are not immunologically silent, as they strongly upregulate both interferon beta and IL-6 production. And something that will become important for the rest of the talk is that these LNP formulated mRNA vaccines resemble STING agonists in terms of their ability to trigger interferon and IL-6 production by macrophages. Now, I mentioned before that one has to be careful that too much interferon can lead to lower production of the antigen encoded by the mRNA vaccine. To monitor that, we have developed a very simple assay in which we use the properties of the F-look mRNA, which is immunologically silent, and the RBD mRNA, which is immunologically active. So what we can do, we can treat mice with the firefly luciferase mRNA alone or co-administered with the RBD mRNA, and then simply do a BLI scan at different time points after administration, in this case, 6 hours and 24 hours. The results are very obvious. You can see the upper row that if you just inject the F-look construct, you have very robust production of F-look, and data are quantified here on the right. You see it mainly in the liver, and then that's basically kind of like where kind of these particles, most of them end. However, if you co-administer the RBD construct, which is immunologically active, that leads to a very strong suppression of the firefly luciferase signaling, indicating that that the inhibition of translation has occurred. Now, to summarize this part, because we think because of interferon, high levels of interferon production, inhibition of mRNA translation can occur, and indicating that potentially reducing but not abolishing type 1 interferon production will lead to better priming of T-cell responses by mRNA vaccines. I'm going to, in the second part of the talk, I'm going to shift gears and talk about these complex metabolic alterations, signaling transcriptional and metabolic alterations that occur following mRNA vaccine administrations, because we feel that this is an area that is, at this point, not at all kind of like developed, and many lessons can be learned if we can map these responses better. Now, the clues that we should be looking at metabolic changes come from clinical data. These are PET scans, FDG PET scans that Jeremy and Johannes, our collaborators at UCLA, have provided us. And it's now kind of a well-known kind of like issue in the imaging field that many patients who have been vaccinated with these two mRNA vaccines, and they undergo an FDG PET scan for a completely different indication, most of the time cancer, they have this kind of very strongly reactive lymph nodes, regional lymph nodes that are on the site of where the vaccine was administered. There are 4 examples here. This creates significant issues because just simply looking at these PET scans will not really tell you whether kind of like this is actually an immune response or cancer, and it occurs, according to Johannes, in more than 10% of people who undergo FDG PET scans, even though some of those FDG PET scans are taken weeks after mRNA vaccination. This phenomenon has been now described in the literature, and you might have seen even a New York Times article about it. Now, for those of you who don't think too much about FDG uptake, it's simply FDG, it's a glucose analog and transported via glucose transporters, phosphorylated by enzymes called hexokinase 1 and 2. And that process can be imaged by PET. And usually, when we think about an FDG PET scan, what comes to mind are tumors which are FDG active. Now, for us, one of the questions, long-term questions, is to understand what mechanisms are behind this increased glycolytic activity in regional lymph nodes. following COVID-19 mRNA vaccines. And we have a hypothesis that cytokines triggered by pattern recognition receptor activation may induce these metabolic alterations. And again, we think that the culprit is probably going to turn out to be type 1 interferon. So we wanted to model these effects, and the way that it's quite difficult to image lymph nodes in mice, and we wanted a surrogate of an mRNA vaccine in terms of interferon production, and we turned to this sensor of DNA, cytosolic DNA called STING, which stands for stimulator of interferon genes. Very briefly, STING detects, it's basically downstream of another protein called cGAS, which is activated by cytosolic DNA and produces a second messenger called cyclic GMP, which then activates STING. The outcome of STING activation is production of type 1 interferon, IL-6, and several other cytokines. Now, what we have been using extensively for other projects in the lab, it's a STING agonist that was generated by JSK, is now in clinical trials for cancer, and that can be administered to mice IV. It activates STING, and the outcome shown here in the bottom, we see very high levels of interferon and IL-6 in plasma, and also in the spleen, we see both cytokines being produced. So now we have a surrogate marker for mRNA vaccines that induce interferon, and we can actually do an FDG PET scan before and after treatment with a systemic STING agonist. And what we see now is that there is a very strong induction of FDG uptake in the spleen, which is very similar to what has been observed clinically in the lymph nodes, in the regional lymph nodes following the mRNA vaccines. And that can be quantified here. Now, what we can do now, we can actually ask the question, what immune cell types in the spleen are responsible for this increased FDG uptake? And the way that we have approached it is to take mice that have been treated with vehicle or a STING agonist. 6 hours later, we collect the spleens and we perform single-cell RNA-seq to identify cell types that have been activated by the STING agonist and look at transcriptional changes that occur in those cell types. So what we see here is this TSNE plot. We see that we can identify the majority, actually pretty much all the major immune cell types in the spleen. This is a STING agonist in the top left part, the vehicle in the bottom. And if you do it with something else that doesn't induce major changes, you'll see these clusters overlapping. But here there is definitely a very strong impact on the transcriptome of many immune cell lineages in the spleen. So to my surprise, I didn't actually expect this. I was expecting that it will be macrophages that will be responsible for this the increase in FDG uptake, but it seems to be T cells and to some extent a subset of B cells, probably germinal center B cells. So what we are looking at the STING agonist on the top, vehicle on the bottom, and a marker of glycolysis, which is HIF-1 alpha, which is strongly upregulated by the STING agonist. Downstream of HIF-1 alpha, there is hexokinase 1, which, as I showed you before, is responsible for the retention of FDG in tissues. And again, it's T cells that primarily in the spleen upregulate hexokinase 1. The second observation that we've made was that there are these markers of T cell activation that people in the immuno-oncology field look at all the time. So we see CD69, granzyme B being upregulated. MYC is strongly upregulated in T cells, and some of these T cells start making a type 2 interferon in response to a STING agonist. However, and surprisingly, we actually see that other markers that are usually associated with more exhausted T cell phenotypes are also upregulated, and those are shown here, like EOMS, which is upregulated in the absence of TBET upregulation. PD-L1 goes up even on T cells, and we see some evidence of caspase-8. The third observation was that T cells and B cells, but especially T cells, upregulate strongly interferon stimulated genes, and that's shown here. Just a few examples, some of the genes that we study in the lab, like PART9 and SEMHD1, all of them prototypic ISGs, are strongly upregulated in the T cell compartment. So what does it mean overall? It's kind of like trying to put it in a context. Looking in the literature, this resembles a phenomenon described before that is called out-of-sequence interferon signaling. So essentially what that means, you can see it here in the left part of the slide. If a T cell will see interferon first in the absence of strong TCR activation, then the response to interferon is mainly transduced through STAT1, STAT2 homodimers, and that resembles an antiviral response in a different cell type, the outcome being anti-proliferative and pro-apoptotic effects. If on the other hand, a T cell will be activated first, especially by foreign antigen, and then they see interferon, maybe at the same time or later, then the response is mainly mediated through STAT4, not STAT1 and STAT2. And the outcome is completely different. Now T cells can differentiate, can proliferate, and these signals promote survival. So to summarize, what we have found so far is that mRNA vaccines can paradoxically sometimes induce too much interferon that can inhibit mRNA translation, and also can trigger these complex signaling, transcriptional, and metabolic alterations, which are poorly understood, but can probably profoundly impact the quality of CD4 and CD8 mediated immunity. So the objectives will be in the future to tune the production of type 1 interferons, and we think that can actually lead to significant improvement in CD8 T-cell responses via multiple mechanisms. So the way that we plan to do that is to decouple the antigen and adjuvant component of mRNA vaccines. That will allow us to tune the strengths of interferon signaling. And the model is essentially saying that if you don't have any interferon signaling, then you are not going to get CD8 T-cell immunity. There is an optimal amount of interferon signaling, and too much interferon will turn out to be immunosuppressive and will, of course, lead to very high reactogenicity. We are also looking at approaches that can actually allow us to uncouple temporarily the two components of mRNA vaccines, and that can be also accomplished. The other direction that we feel it's very important is to continue to optimize the PK properties of these lipid nanoparticles. At this point, the majority of them end up in the liver. They were generated, many of them, to promote expression of various proteins in the liver, and simply there was not enough time for the field to tweak them significantly to abolish that effect. So there is uptake in the spleen. We would like to see much more uptake in the spleen and much less uptake in the liver. And there are already kind of some solutions to this developed by BioNTech, but those are not really kind of accessible to us at this point. So groups at UCLA working on lipid nanoparticles will be extremely helpful to fix this problem. And we want to leverage more the PET imaging probes for immune activation developed at UCLA, including 18F-clofarabine, that something that we have developed a few years ago and took to the clinic in collaboration with Dr. Chernin. And as you can see here, we can image in healthy volunteers lymph nodes, which I think it's the only PET probe that can accomplish that in somebody who doesn't have an immune response. But the goal here will be to see if we can see even higher activation of increase in lymph node uptake of these probes. And overall, kind of, we think that it's important for UCLA to continue to expand the mRNA vaccine program, given that this is an area of very high significance, not only in viral infections, but also in cancer. With that, I want to acknowledge the people who helped with the project. And in my lab, it was Khalid Rashid, a postdoc working with Doug, Evan, and some outstanding rotating students, Haley, Daniel, and Caitlin. In Ting-Ting's lab, Alex Lam, and you'll see a lot of Alex's work in Ting-Ting's presentation. I want to thank the UCLA TDG and Dr. Steve Smale for helping with the agreement with Acuitas. That was actually difficult to set up. It took actually probably 3 months to get it going. And our collaborators at Acuitas, especially their Chief Scientific Officer, Ying Tam. And finally, very important, this work will not have been possible without the COVID-19 award that my lab and Ting-Ting's lab received in May. So with that, I'm going to, I don't know if we want to have questions now, maybe at the end will be better. So maybe Ting-Ting, you can take over.
Yeah, Ting-Ting, do you want to show your presentation? That was really great talk, Caius. I think there are lots of questions, but because of the interest of time, let's keep all the questions for the end. So. Oh, Ting-Ting, I think you're muted.
Okay.
There you
Can
go.
you see it?
Yep, we can see it.
Let me find my pointer. Okay. So Caius has told you about the basis of and the history of the mRNA vaccine. So I'm going to give you a brief overview of the data we generate based on the construct we obtained in October, last October. So prior to COVID-19 pandemic, our mRNA vaccine had been tested for several emerging infectious diseases like Zika, Ebola, and avian flu viruses. And all these studies focus on antibody. And it's clear that these mRNA vaccines are very efficient at inducing antibodies. So on the contrary, the CD8 T-cell response from these nucleoside modified mRNA vaccine are variable. So this is our one objective, and to see whether we can tune the property of this mRNA vaccine to maximize their capacity of inducing CD8 T-cell response. But before that, we first do some research experiments on the mRNA vaccine construct we received. And here are the two mRNA constructs we synthesized for the project. This is already shown by Caius' presentation. The SARS-CoV-2 RBD construct is fused with this T4 Fabritin trimerization domain for multivalent display. And this is a very similar design to the first generation of Pfizer vaccine. Now the one we have, it's full length, but the one we generate for this project is just RBD. And then also we have this signal secretory peptides at the N terminal. So the RBD can traffic through the ER and is secreted outside of the cells to bind to B cell receptor. The other construct is a full-length HA hemagglutinin construct from the influenza virus and has this point mutation to stabilize its perfusion conformation that you pretty often heard about. The spike protein vaccine we have, the mRNA vaccine we have, they had this mutation to stabilize their perfusion conformation. So we got these mRNA lipid nanoparticle in mid-October, and then did the first injection in November. And in the first experiment, we inject mRNA-containing particle intramuscularly, and we sectomize around 6 weeks to examine antibody and T-cell response. And here we pour serum from multiple mice to examine the antibody response. And on the left is antibody titer determined by ELISA. We include the serum from the flu-infected mice that we also collect at 6 weeks post-infection. And then here it's just one injection. And then the one injection stimulate generate antibody comparable to infection. At the right is a hemagglutinin inhibition assays that determines the amount of a neutralizing antibody in the serum. And as you can see that also one injection of a low dose, like 2.5 microgram, could induce neutralizing antibody only slightly less than natural infection. And then of course, a 25 microgram give you more neutralizing antibody. And this is expected. Basically, the data show that these mRNA particle, lipid nanoparticle are very immunogenic. So how about the T-cell response? Now, we expect to see a low level of a T-cell response at 6 weeks post-immunization. So we use the ELISpot to quantify the interferon gamma secreting cells upon antigen stimulation. So under the bottom of the graph, the image from the individual wells. And with the stimulation of whole inactivated virions, this is from flu, you can see some interferon gamma secreting T cells, and those are from the CD4 T cell response. Although this response are quite variable among mice, we did not see much difference between the high dose and the low dose. Now, the CD4 memory response are also pretty comparable to those generated by infection. Shown here, this is only one mouse from the 6 weeks post-infection. And then on the right, the result from stimulation with a peptide corresponding to a CD8 epitope in HA. And the response were also comparable between high and low doses. So we also examined T cells in the lung. Now for respiratory viruses, mucosal memory response will be beneficial. And then interestingly, we did detect some cellular response in the lung. They were pretty weak and compared to the infection, but they are there. And at this moment, we don't really know whether these are resident memory cells or circulating effector memory cells. And more experiments are needed to distinguish this population. So similarly, with RBD mRNA vaccine construct, we also observe cellular response in both the spleen and lung. Now here we use a peptide mix derived from the RBD domain, and to re-stimulate T cells. By depletion, we know that 1/3 of this response is from the CD8 T cells. So we then ask whether the cellular response could be increased by 2 injections, as well as with a higher dosage of mRNA vaccine. In this experiment, we use the RBD construct. So the second injection indeed significantly increased the antibody titer as shown here, about 30-fold higher for 2.5 microgram and about 10-fold higher for 25 microgram. And also the higher dosage of mRNA vaccine definitely induce a higher amount of antibody, which is consistent with the published literature. And Dr. Muscami's lab also help us determine that this serum contain high level of a neutralizing antibody for SARS-CoV-2. So 2 injection and then also significantly increase the amount of the T cells in the spleen as well as in the lung. Now, similar to 1 injection, a 10-fold higher dosage did not really enhance the cellular response. Now, using a peptide derived from a known CD8 epitope in the RBD to re-stimulate T cells, we also detect a CD8 T cell response after 2 injections in both the spleen and the lung. And then this CD8 response was almost undetectable with only one injection. So we showed that intramuscular injection of a nucleoside modified unpurified mRNA lipid nanoparticle are capable of inducing CD8 T-cell response in the spleen as well as in the lung. And then this may not be a surprise since unlike most of a protein-based vaccine, mRNA vaccine have de novo protein synthesis, which enable MHC class 1 presentation by dendritic cell to prime CD8 T cells. Now we do need to further characterize those T cells in the lung, whether they are circulating infected memory cell or tissue resident memory cells. This will be a very important property to have for respiratory virus vaccine. So if a systemic administration like intramuscular injection could generate mucosal cellular memory response, that will provide immediate action to control infection upon rechallenge. Now we showed that both in the case of HA and RBD, a 10-fold higher dose of the mRNA vaccine increased antibody response, but did not significantly impact T-cell response. Now a second injection increased the T-cell response by 10-fold. So this nucleoside modified mRNA was synthesized to reduce their immune stimulating ability in order to maximize the protein expression as described by Caius. But we found out that these mRNA are not complete immunological silence. It's pretty active on the contrary, and which can provide some adjuvant activity to drive activation and maturation of the dendritic cells. Now, balancing these two features of mRNA vaccine, protein expression and immune stimulating, is most likely needed to maximize their capacity of inducing CD8 T-cell response. Now, in order to investigate the potential of mRNA vaccine to stimulate a robust, protective T-cell response, we need to have an infection model that depends on T-cells for protection. And before I tell you the model that we propose to test, I want to give you just 2 slides about what we know about T cells and the SARS-CoV-2. Now here is a working model for COVID-19 immunology and the disease severity from this very excellent review by Shane Crowdy and Alessandro Setti. So in this model, the disease severity is due to the viral invasion of an innate immune response, probably type 1 interferon response, leading to very high viral loads and a lack of a timing T-cell response, culminating in hyperinflammation and lung immune pathology. Now, previous studies have shown that the correlation of both CD4 and CD8 T-cell response with the reduced disease severity in primary SARS-CoV-2 infection. Now, this result indicates that like most other virus infection, T-cells are crucial to control the infection and stop patients from progressing to severe disease. Now, in this UK study, about 2,600 essential workers were recruited to analyze their T-cell and antibody response to SARS-CoV-2. Now, based on the number of T-cells in ELISpot assay, they were divided into 2 groups, high and low, and also positive people in the high group. Now, these people were then followed for about 3 months, and 20 of them developed symptomatic COVID-19 disease confirmed by RT-PCR. And all of those 20 people were in the lower T cells group. Now, although the infection number is low, this study supports the protective role of a T cell against infection. Several studies had also mapped the T cell response and shown that in addition to spike, membrane protein and a nucleic protein, nucleic capsid protein are also the target for T cell response. Typically, each person can generate T cell that targets multiple regions of the protein. So this stimulates the interest of a developed T cell based vaccine that could be more tolerant to the emergence of a mutation escaping the antibody. Now I'm going to move to the model we want to use to test the T-cell, to test the ability of mRNA vaccine to provide T-cell mediated protection. Now, prior to COVID-19 pandemic, most of attention were on flu. As shown here, it's estimated that flu caused 24,000 deaths in the US in the previous season. Now, as many of you are aware, flu cases are way down this season. Now, vaccine efficacy for flu vaccine remain far from optimal, but still is the best way to prevent flu-related illness. Now, current seasonal flu vaccine are based on injectable subunits or inactivated variants, which primarily induce neutralizing antibodies specific for HA. Now, high mutation rate in the global region of HA can severely compromise the protective capacity of a vaccination-induced antibody. So this emphasizes the need to develop new flu vaccine capable of mediating protection against a broad range of strains. Now, broadly protective vaccine are defined as a vaccine that cover a large subset of influenza virus. Now, to design this type of vaccine, conserved protein of the virus are targets. Now, current strategy for such flu vaccine mainly focus on generating the broadly protected antibody direct against this conserved but immune subdominant region, like the stalk region of HA. Now, the MP and the M1 are both important structural protein of the virus that are highly conserved across the subtypes. They contain a strong T-cell epitope. Now, in the absence of a neutralizing antibody, heterosubtypic protection has been shown to be predominantly mediated by CD8 T-cells in numerous animal studies, although evidence in humans was limited. Now, two separate studies were carried out during 2009 pandemic support that people with a higher level of a pre-existing CD8 T-cell recognizing highly conserved epitope had lower risk of a virus shedding and experience mild symptoms. And moreover, in the animal study, cross protection is associated with the homing capacity of a CD8 T-cell to the lung. Now, since we see CD8 in the lungs after injecting mRNA vaccine intramuscularly, we want to test whether mRNA vaccine based on conserved flu protein can provide T-cell based heterosubtypic protection. So in this paper, it was shown that using adenovirus to induce memory T-cells specific to a highly conserved protein, nuclear protein, could provide protection against lethal flu challenge from different types. So on the top was the viral titer results consistent with the role of a memory T-cell. They don't prevent infection, but could facilitate the clearance of the viral infection. Now, this benefit led to the survival of the mice and reduced weight loss shown here. Therefore, we will use the MP protein and a flu infection model to test whether mRNA vaccine is capable of inducing protective T-cell response. This is our current design for MP mRNA vaccine. Dr. Yuan Shi compiled all MP sequences from influenza A viruses in the database and derived a consensus sequence. So we use this sequence to generate mRNA vaccine. In addition, to facilitate the presentation of the MP, the MHC class 1 signal peptide and the trafficking domain were added on the sequence. So in collaboration with Caius group, we will test the timing and the type of adjuvant to maximize the ability of the mRNA vaccine to induce T-cell response. Now here is our plan for a future study. we will examine DC activation and T-cell response, and then challenge with lethal doses of the influenza virus. And then hope the result from this study will help us to generate a better mRNA vaccine that could induce a more robust CD8 T-cell response, not only in the lymphoid organ, as well as in the peripheral tissue. Now, finally, I would like to acknowledge the people who did the work. It has been a wonderful collaboration with Dr. Caius' group in this project. Special thanks to Alex in my group and Khalid and Evans in Caius' lab. And I had to pull Alex from his thesis project to carry out all this immune mouse study. Patricia and Benencio are my rotation students who help with the ELISA data. I also want to thank all the UCLA collaborators, Dr. Sun and Dr. Shi, for their expertise in influenza virus, and then Dr. Wami's lab, who is the go-to person for SARS-CoV-2 at UCLA. And of course, without the funding from the research, COVID-19 researcher, we would not embark at this journey. And we learned so much about, we heard so much about mRNA vaccine. So it's really an excitement for us to actually work on that. And then when we actually started working on that, we realized there is still a lot we did not know about how they work. And then only until we learn more about it, we can maximize its potential for infectious disease as well as for cancers. All right, thank you.
Great, thank you so much for really stimulating talks and really complimentary. So I guess we'll open the floor up for questions. There was already some discussion going on. And I know there was an earlier question that is more for the clinical folks here, but maybe the speakers can also comment on that. And then I guess, and then Feng, will get to you in a sec. So the question, earlier question about, can you discuss what interferon reducing mRNA transcription really means? Does it mean that less or more vaccine response? And what does it mean for patients infected with the native virus as early? And sustained type 1 interferon production seems to be associated with mild disease and late, and slow interferon is associated with severe disease. There's a little bit of a contradictory kind of phenotypes, right? Also thinking about the interferon response for the pathogenesis side of things. If you guys would like to comment on that.
So maybe I'll start. So essentially, there is probably a very narrow temporal window in which a type 1 interferon response can protect against the virus. However, kind of like if now interferon levels actually are very high late, then that has been shown to be very actually detrimental. Maybe the mechanisms involved are at least in part associated with T-cell dysfunction that has been shown both in viral infections and in cancer being associated with interferon signaling at the wrong time. Now, what it means in terms of too much interferon for the vaccine, I think that if there is too much interferon, then there is a risk of shutting down prematurely the production of the antigen. And we actually see that in cell culture. In fact, sometimes like in some of the assays, the level of interferon is so high that we see a global repression of transcription with mRNA levels for, essentially going down. So it will be, again, no interferon is bad. Too much interferon is bad by a different mechanism. It has to be tuned to be in this kind of like range in which interferon can play positive roles without having the detrimental effects.
Great, thank you. No, I think the timing is very tricky to have the perfect response, both early and later. Ting-Ting, do you want to add anything or happy?
No, I think yeah, that's you already talked about that. The timing is really critical, and I think there are some studies saying that if you give the interferon ahead of the time, then you can prevent the disease progression. But if you give like 1 day or 2 days after the virus already starts, you can actually make it worse. So it's like, you know, it's very tricky to for the interferon.
Yeah, I agree, Ting-Ting. And I think, you know, when we do mouse studies, you can actually really time it, right?
Yeah.
Specifically, and you really see sort of the phenotype, sort of day and night, sometimes within a really short time period. So, okay, so let's go to Feng first, and then we'll go back to the chat for questions.
Okay, well, great talk. So I have 2 questions. The first one regarding the RNA used in the vaccine. Does it contain the double-stranded DNA template, or is it transcribed using the RNA polymerase mutant that Moderna developed that minimize the double-stranded RNA produced?
I think that the amount of double-stranded RNA can be reduced by various approaches, but without HPLC purification, or there is another approach, cellulose purification will not be eliminated. So, and then depending on the dose, as you know, the Moderna vaccine, it's actually kind of 100 micrograms. That's a pretty high dose, and it's going to be very difficult to actually kind of not have any double-stranded RNA. And not having any will be equally bad, because then it's already modified, so it doesn't activate TLR7 and 8. And if it doesn't activate any sensor, any RNA sensor, then you don't have the adjuvant effect. So that's the challenge here, because you have like both components of a vaccine encoded in the same kind of like essentially, you know, kind of like molecule in a sense, yeah.
Is the DNA removed?
The DNA is removed, yes, yeah.
Okay, okay. My second question is that because the protein, recombinant protein generated by the vaccine is secreted, is the protein glycosylated? Is that known? Because the secretion pathway sometimes add a glycosylation site.
Yeah, so if it's if they contain a glycosylation site, it will be glycosylated.
Well, it's I guess it's not typically secreted, right? So I guess could there be accidental?
Well, I think that the HA, for example, if you are talking about HA and spike, they are membrane anchor, but they still go through the ER, Golgi, and they get glycosylated there. But they were just like anchored to the membrane. They don't necessarily secrete it.
I see. The same true for the SARS-CoV-2?
Yeah, the spike is it's the case too. But I mean, if the cells die, they release the protein, and then the protein will be released outside the cell. But most of the time in the infection, they are on the membrane.
Cool. Thanks very much for the answer.
Great. And I think Caius addressed the question about the storage improvements. And the next question is, do you have any prediction on whether the memory CD8 T-cells in the lungs are resident or circulating? That's for Ting-Ting.
I mean, of course we hope to be resident memory T cells, but they really need to do a little bit more study to characterize them. But based on the level in the spleen, we think that unlikely all of the T cell we see in the lung coming from the circulating effector memory cells, some will be resident memory cell. But again, we will have to do more experiments to determine that. And this is going to be very interesting, especially for the respiratory vaccine, that having a mucosal T cell response is really good for that.
Cool. I think I see another person unmuting. Is there any other question? Okay, maybe just an accident. So I have a question. So I mean, I think it's all very fascinating, and I really enjoy all the mechanism and all the all the exciting new data. I guess my question is that it's really interesting that the CD8 T cell seems to be the problem, right? But the CD4 seems fine. So thinking about the CD8 T cell response, you really need both the virally infected cells and the cross presentation from the dendritic cells, right? So I guess what I'm curious about is exactly where is the interferon acting on, right? Because in Caius model, that's really nice. It seems like it's really the dendritic cells that take up the mRNA vaccine. Then you have to make the proteins, and that's where, you know, as me and Ting-Ting know, a lot of the ISGs we study, right, target either mRNA degradation or translational shutdown. So that's where you think that will be happening. But what about, let's say, the muscular cells that also can take up the lipoproteins? Would they have problem also presenting, and sort of like what is the relative contributions of those different mechanisms to the T cell, the CD8 response?
Caius, you want to jump into it?
It's a great question. I mean,
Yeah.
Ting-Ting keeps saying that we have to look at the site of injection, we haven't done that. I think that there will be major changes there too. You have seen maybe on the PET scans that you can actually see the injection site lighting up with FDG as well and persisting. But I think there will be effects of interferon on in the case of IM administration in the lymph node, and they will impact not only antigen-specific T cells but also naive T cells. So and those are really, as you point, those effects are not really well understood. Definitely, Ting-Ting showed you that there is a T cell response, CD8 T cell response in mice, but in humans, at least with the Mrna vaccines, those effects have not been detected. And there is a discrepancy, therefore, between mice and humans that we don't understand. And also, if you look at the dose, it doesn't scale neatly, so from mice to humans. There are quite a few papers where people use like 10, 20 micrograms per mouse. And the mouse is typically 20 grams, and not very different amounts are used in a 70 kilogram human. So again, we don't understand if kind of the dose differences between mice and humans can actually impact responses. Maybe, you know, that can be one factor.
Yeah, no, that's a really good point. And I also noticed in Ting-Ting's data, it's really interesting that with the highest dose, among the two mice, yeah, you're curious, there's also huge diversity or the heterogeneous.
Yeah, it's a because I think one of the reason is that the T cell response we are looking at is like 6 weeks post immunization, so they are pretty low. But if you look the two doses, even though there is variation, you don't really see big difference between the low dose versus the high dose. And that is something actually kind of surprising to me. Maybe it's for CD8 T cell, maybe you don't need a lot of the antigen, you just need to get some amount of antigen, and then you have a good maturation of the dendritic cells, and then that it's sufficient to prime the CD8 T cell response. But the variation indeed was really big. And then I think one of the things that we had to go back to do is to look at early time point, like maybe 7 days to 10 days after the boosting. That way you can see the more like the peak of the T cell response, and maybe that will give you more consistent number rather than variation we were seeing here.
But then also, Ting-Ting, I guess even if the response is not really different, you know, going, you know, in the same direction as the dose, I think about for the memory population. I mean, I'm no immunologist, but I know the debate is always that, do you need antigen, and how much antigen you need to maintain that memory population? So could that also have maybe long-term consequences on the memory cells, how much you give initially?
Well, I mean, It's the amount, I think a few study previously already shown that the amount of antigen expressed in the beginning affect the peak of the response, but doesn't really affect the ultimate pool of a memory T cells. And then or their function of T cell or their function. So I think there is the requirement for CD8 T cell is much lower compared to CD4 T cells. And then we had some study that you don't even, as you know, we do a lot of live attenuated virus that we don't even see any viral replication whatsoever. And yet the CD8 T cell response just as good as wild type. It's even better. So, so I think that there is a different requirement in terms of the CD8, CD4, is that when you do the presentation and then priming to T cells, the environment, the molecule and then everything.
So maybe less is more for CD8.
Yeah. I mean, that's a really interesting point you made about the attenuated virus, right? What if that takes away the interferon side of things, the bad stuff from the interferon? Yeah. Anyways, Feng, do you have another question?
Yeah. So I'd like to ask the speakers about to address the vaccine escaping mutant of viruses, finding the constant region is one. How practical it is to include the multiple variant in a single vaccine? That's my question.
Maybe I can start. I mean, there are two approaches. One, I think that now they are just including one kind of mRNA, variant per vaccine. But we know that Drew Weissman, who wants to actually kind of have 14 or 20 different mRNAs, not only for one virus, but for essentially kind of immunize against multiple viruses in a single vaccine. So I don't think there will be in the long term a limit to how much. I mean, of course, there will be an upper limit, but you can easily probably kind of have multiple mRNAs there. In fact, at least in the cancer field, the way that you will use the mRNA vaccine for cancer, you always have multiple epitopes encoded in that case in a single mRNA, because you kind of use shorter epitopes. But that's a very good question. There are multiple ways of addressing it, and as well as what Ting-Ting presented before, kind of including other genes, viral genes that are subjected to less kind of mutations, and that's an approach that is being now looked at.
Thank you.
Great. And if we don't have any more questions, I think we've exhausted our speakers. Thank you so much for joining us today. I think we're all very stimulated with the discussion. So bye-bye, everybody. Have a great weekend.
Bye.
Thank you. Bye-bye.
Bye.